Record. Okay. Yeah. Okay. So, hi everyone. My name is Momo Arai. I'm a fifth-year medical student, and today I'll be covering pathology, pathology lecture, talking about tissue repair. Now, there's two lectures, but like I tried to combine them and only focused on what like I'm covering everything for sure, but I'm also focusing on important things that he can ask you in the exam and so on. And these two are related, so we'll go through the first one and the second one. If you guys have any questions or if you want to correct me at any point, just like you can type it in the chat or unmute yourself. I don't mind at all. Okay. Okay, so let's start. So this is what we'll be covering. And again, tissue, whenever you hear the term tissue repair, it's from the name itself, it's to repair any tissue that's damaged, right? Now, when do we need this process to happen? We need that process to happen whenever there's inflammation. I'm sure there was an inflammation lecture before this, but basically inflammation is any sort of trauma or injury or anything that disturbs the cell, okay? Or part of the like tissues and in general. So inflammation, if you have like an injury, let's say a cut or any sort of injury to the body, there are three sort of like pathways it can go, like our body can go through. So it's either the cells. This is, by the way, a summary. I put this picture from your summary slide, but if you don't like, this is like in general, in summary, okay? We'll be covering this in more detail, but as long as you get this idea by the end of the lecture, then that's really good. So yeah, so again, like I said, if there's any inflammation, there's any injury, your cells can either regrow like back to normal state 100%. That's called regeneration. So your body will recover basically, and it will be like as if there was no injury. So it will function normally as before. That's the first outcome. The second outcome is the cell, if so we'll cover this part later, but basically if the injury is too much or if the cells themselves are not capable of going back to the normal state, it will repair. So I know repair is kind of like the whole lecture is called tissue repair, but it's a general term. We'll get into more. There's a difference in like terminology. Like whenever they say repair versus this one regeneration, it's different terms. So again, they will repair, meaning that they will not go back to its original like function, not completely, but they will overcome that kind of damage by forming a scar. And you guys know this by like in it, like it's general knowledge that, for example, if you cut yourself, if the cut is too much, for example, you'll get a scar, right, or a scab. So if you imagine it like that, that's the second outcome. Now the third one is if the damage is too much for the cell to kind of like recover from it at all, it will undergo chronic inflammation, which is inflammation over a long term. Now these three outcomes is like is what happens whenever you have any sort of injury to your body. And we'll cover this, like the repair regeneration part in detail. And we'll also see why does some cells, for example, not recover, or why do they undergo chronic inflammation and so on, and the factors that go to this side, okay? So we'll try to cover all of that like today. Now, so a better term for this lecture, I would say, is healing, because again, I don't want you to get confused with the terminology when I go into it. So healing, again, if there's any sort of injury, your body can go undergo two processes to process this injury. So to kind of overcome this injury, they can undergo regeneration. Again, it's like recovery. You can try to memorize that as that. So recovery means that it will go the cells will be restored to the normal function, like in their natural state without any sort of like scar. So it's we call it scarless or no scar tissue. Okay, that's regeneration, or again recovery. Whenever we say repair, that means that like it does go back. It's able to process that injury, but with a scar formation. Okay, so we will see like a new tissue is formed to kind of compensate for that injury, to kind of cover it. But they're both they fall under healing. Okay, is that clear for everyone? I just wanted to start with this because to have a good like basis before we go on. Okay. Okay, so we talked about regeneration and repair. These are both kind of how our body processes the injury. Okay, now what will determine if let's say this cell will undergo regeneration, like completely back to normal or partially back to normal? This will depend on, which I'll cover this in detail later, but it will depend on the cell type. Like I said, is the cell itself able to go back to normal? Is it able to like give it partial function after the injury or not? Okay, so it depends on the cell itself. Then it depends on the amount of damage that occurred. So it makes sense from the name, but amount of damage, like the more the injury is, it's harder to recover, so it will likely undergo repair than regeneration. Do you get the idea? So it's the cell itself, the amount of damage that occurred, and the surrounding the cell, what we have? We have kind of like a support around them, right? It's like a support system for this process to happen, which includes things like growth factors and matrix, extracellular matrix. ECM stands for that. Now, again, we said cell type, amount of damage, and any other factor that's affecting like the vicinity of the cell. Why is that? So all these factors will determine if they undergo whether regeneration or repair. And this is important to understand. Like this basis is like what the important thing to understand before we move on and talk about everything, like every one of these points. Why is that? Because regeneration and repair, basically healing in all generally, will involve the cells, of course, that was injured, and it will involve interaction between the cells, like the cells here, and around it, extracellular matrix. Okay, that's why I put that as support, and it's very important. Now, this picture, he put it just to show you, he put a lot of pictures in his slides. The most important things to know are the patho images, because they can come up in like in your exam, in OSCE and so on, OSPE, sorry. But this is just to kind of show you the same thing, regeneration versus repair. You can see that regeneration is like completely back to the normal. It's completely the same. Repair is, there is kind of like a scar tissue. There's new things going on, which I'll get into in a minute. Okay, so I hope that's clear. We'll talk about all of these things. This is the bulk of the first lecture. Now, okay, wait. So yeah, so this is the entire thing of the first lecture. We will talk about cellular proliferation. Before we get into, so this will talk about the three main points that I talked about just now. And before we begin, he talked a bit about stem cells and what cells are involved. So I'll try to touch upon that a bit because he wants you guys to understand before you know like the type of cells, like where they come from and so on. So again, like I said, healing in general, involves cellular proliferation. Okay, of what type of like tissues or cells? The injured part, it will involve like blood vessels, and it will involve fibroblasts, which is again in the extracellular matrix. So everything together. So the cell itself and the surrounding area. Now this, like he put this picture to show you, again, I'll get into stem cells. So this is why like he just put this, and it's for you guys to understand that there are stem cells, which are cells that can give rise to more cells. Basically, that's the meaning of stem cells. Okay, and it will give rise to more cells. It can differentiate then, like from here, it can differentiate into, let's say, into specific functions, specific cells, or it can give rise to more of its own kind. This is called proliferation. Or it can die if there's any damage, or if the lifespan of the cell like was reached or so on. So that's called cell death. Okay, so this is just how what a cell can undergo normally. And then we'll talk about what happens in injury. So again, see here, this is everything that's involved in healing. Okay, we said cells, you need, again, interactions, right, between cells and the surroundings. So cells, growth factors are kind of like signals for interaction. So cells, growth factors, you need receptors to kind of take that growth factor or like process growth factor in terms of signaling. And you need the extracellular matrix or support. Does that make sense? Like you have the cells, you have the support matrix, and you have interaction between those two in terms of growth factors and receptors to receive that. Okay? Does that make sense for everyone so far? Because I'll get into it, like I'll talk more about it in details, like don't worry. Okay, good. Okay. Now, okay, so this is where I was supposed to ask you guys, but okay. Now we'll move on to stem cells. Stem cells, again, like I said, there are cells that can give rise to more cells, either their own or like specialize into different cells. There are two types of stem cells that he talks about in this lecture, and that is important to understand, is embryonic, or sometimes called pluripotent stem cells, and adult or somatic stem cells, okay? Now, there is a better picture to kind of like understand all of this, but basically, early in life, like in the embryonic stage, there is there is a state called blastocyst. Basically, at this point, like it's a group of cells that can give rise to everything in your body, every cell you can think of in your body, and these are called the embryonic stem cells. If you take the inner like mass of the blastocyst, which is the group of cells, these are called embryonic stem cells. Now they're also called what? Pluripotent, we said. And you can see here in this side, because so pluripotent is kind of like the state of the stem cell. So whenever they say embryonic or pluripotent, they mean the same thing. It can give rise to basically all types of cell in the body. So that is embryonic or pluripotent stem cells. You see it's developed from the inner cell mass of the blastocyst or the group of cells, and they can give rise to all tissues in the body. And then, okay, so the next thing, the point is here. So pluripotent, these stem cells can further give you so okay, this is kind of confusing, but it can give you multipotent stem cells. Multipotent means that it can give rise to only a specific lineage or specific type of cell. Let's say you've heard about hematopoietic stem cells, right? In like foundation and like heme. So hematopoietic stem cells, or let's say mesenchymal stem cells, like these stem cells only give rise to their own kind. So hematopoietic to its own kind, which is blood cells, like red blood cells, white blood cells, and so on. So that's the example that's shown here, but there are other types of multipotent stem cells. Now, why did I say like it's confusing? Because it tells adult here, right? So in here, sorry, here we also talked about adult stem cells. So yes, so these are kind of related because pluripotent stem cells, like these stem cells, can give rise to adult stem cells. Like we said, pluripotent gives rise to any cell you can think of, including the adult stem cells or somatic stem cells. These are just different names for it, but you get the idea. So like follow this picture and it will make sense, like what was written on the slide. And wait, let me go back here. See, it says it's more restricted, meaning that it can only form its own type of cell. Okay, so that's the difference between embryonic / pluripotent versus adult / somatic stem cells. Now, why are we talking about stem cells in the first place? It's because he wants you to say that there are cells that can proliferate and make up for the injury, which we'll get to in a minute. So stem cells, we have embryonic, and this is, again, adult stem cells, cells that can give like its own kind, like proliferate again and regenerates its own cell or differentiate. Okay, that's the definition of stem cells. And then this part, we said embryonic, pluripotent give rise to everything. And then it even gives rise to the adult or multipotent stem cells, which can give rise to its own sort of cell type, which with its own specific function. Okay. Now, there are some examples of adult somatic stem cells he puts in the slide, and these are some of them. So, for example, hair cells, right? Hair cells, they can give rise to they will like regenerate like every in a certain time, right? So there are stem cells for hair cells, which is found within the hair follicle itself. And again, you don't need to know this in too much detail. He just meant these as examples. So there are also cells in the liver, for example, that are considered adult somatic stem cells, and cornea. In your eye, there's a part called corneal stem cells, which can give rise in case there's any damage and so on. These with the hematopoietic stem cells are examples of adult stem cells or multipotent stem cells or somatic stem cells. Like all of them mean the same thing, okay? Is that clear so far? You guys don't have any questions? Like I'll move on. The basic like kind of idea I want you to get from that is like stem cells, the two types of stem cells and examples, okay? Now we'll move on to the type of cell. Remember how I said there are the type of cell determines whether it can recover or like regenerate or repair, right? It can undergo repair. Now, tissues of the body, there are like, so these are types of tissues in the body. And there are three types. They're labile cells, stable cells, and permanent cells. So again, we finished stem cell part. These cells will talk about whether they have the ability to kind of regenerate or undergo repair. Okay, so this is the important slide for this, like for the tissue type for this lecture, rather than the stem cells. But again, it's important to have a good understanding of all of it. So okay, so labile cells are cells that can continuously divide and can easily regenerate. See the terminology, it can regenerate. So if an injury happens, let's say the examples are here: bone marrow, skin, and your GI tract, your digestive system. So all of these can, if there's any injury, they can go back to its normal state. Why is that? Because they have the ability to divide. Like we'll see now, because they have a lot of stem cells and they can go through the cell cycle many number of times, okay? Again, do you realize stem cells came up here? That's why it's important. Like there's a lot of stem cells in labile cells. That's why they're able to go back to normal. Stable cells are between labile and permanent. Okay, so stable cells, they can divide, but only if there's like a trigger for it. So labile cells, I forgot to mention, continuously is important. The word continuously. Why is that? Because even in normal circumstances, your skin will like regenerate, right? Every month, every 2 months, like your skin, there will be new skin being replacing your old one. Even bone marrow, like I said, hematopoietic stem cells and GI, your GI tract is also like doing the same thing as your skin. Okay, so these are continuously dividing. Stable cells, on the other hand, can divide if it needs to. Okay, that's a better wording for it. It says limited ability to proliferate and regenerate. So you can kind of guess like it can undergo regeneration. Okay, again, now the second factor will come in, the amount of damage. So if the amount of damage is not too much, these cells can undergo regeneration, which is the first path we talked about. Exception, oh yeah, so there is an exception he talks about, which is the liver. So the liver has a better kind of like better ability to go back to normal rather than kidney, pancreas, mesenchymal. That's all he means. Because and he gives you an example of saying, if for example the liver was let's say in a surgery you had to take the lobe or like part of the liver, it can regenerate. If not much was taken from the liver, it can regenerate. The rest of the cells can proliferate and regenerate to kind of make up for the lobe that was taken away. So this is an example, and we'll see why, because again, there is a state in the cell cycle, which is called G0 phase. So cells of the liver, kidney, like we saw here, like pancreas and mesenchymal cells, they're all kind of in from the name stable. Okay, so they're in a stable state, we can say, or in a phase that's called G0. Okay, so this is the cell cycle. You have cell division, like it goes through G1, S, G2, M. You don't need to know like the details for this lecture, but just know that this is how it goes through, like a cell goes through division, and giving you like two cells at the end, right? So this is what happens normally in the continuously dividing cells, which are called labile cells. Again, examples: skin, hair, GI tract, and so on. The stable cells, this one, example here, it's written, yeah, hepatocytes stand, like they're the same as liver cells, but again, kidney and so on. So if there is an injury, so we said they're in G0. If there is an injury, it will go back into the cycle and kind of divide and give rise to more cells to regenerate, to be able to regenerate. Now, the last thing we have left to cover is permanent cells. Permanent cells are out of the cell cycle. Like they're from once they're formed, in the embryo, like after the baby is born, let's say, they are completely out of the cell cycle and they cannot enter it again. So basically, if there is any injury, they will die and they cannot regenerate. See the terminology? So permanent cells are, some of the examples of permanent cells are things like neurons, your nerve cells. Like once they're damaged, they cannot regain their function, right? And the person will have paralysis after like severe damage. So they cannot proliferate, they cannot regenerate, and they remain in G0, or you can consider them like completely out, like they cannot go back into the cell cycle, basically. And or wait, contain, yeah, this part, I'll remove this part when sending the lecture to you. They don't contain like stem cells. I think it's just copied from this one, that's why. Okay, and examples again, neurons, your heart muscle. So if there's heart attack, let's say, it won't go, like there will be scar formation, right? And even your muscle cells. So these are examples of permanent cells, and I put a picture for scar here to remind you that these will not undergo regeneration. They will undergo the second pathway, which is repair. So they will have scar tissue, and like they cannot go back to its normal state. So that's permanent cells. And here, wait, okay, here in the cell cycle, again, they're outside, like they cannot go back in the cell cycle. To sum up, labile cells always proliferating. They can regenerate easily. Stable cells, again, if the damage isn't too much. Liver is a very important example he likes to ask about. And basically, it is in G0 and it can go again into the cell cycle. Permanent, they cannot go and they cannot regenerate. Okay. Oh, yeah, sure. Stable cells. Oh, yeah. Okay. So here, like I'll go back to the slide, but basically stable cells are between labile cells and permanent cells. Labile, which is continuously proliferating, think of like your skin or your hair cells that are continuously, like every day they can grow, right? Permanent cells, I'll talk about the opposite. Permanent cells are opposite. They cannot at all, such as your nerve cells or your heart tissue and so on. Stable is in between. So stable, from the name, it's stabilized in G0 phase. Okay, it's a phase in the cell cycle where it's just there, like it's not dividing every day, but at the same time, it's also if there is any damage to it, if there is any damage to, for example, liver, the liver or the kidney, it can make up for the damage and go to regeneration. Does that make sense? Like basically, if there is damage, it will act like labile cells. Okay, only when there is damage it can divide. But if there's nothing, it will just stay stable. Yeah. Okay. No worries. I hope it was clear. Like I hope I didn't confuse anyone. So just remember these type of cells. So labile, stable, okay, in case of injury, will undergo regeneration, which is repair, again, which is healing without scar tissue. Okay, it will go back to normal. Permanent cells will have scar tissue. Once there is an injury, it cannot regenerate, so it will form a scar. And we'll see how that happens in a bit. So these are the type of cells. And then we also said, okay, so we said cells were the first thing. So it depends if they're labile, stable, or permanent, if they can undergo regeneration or repair, right? The second thing is the interaction between the cells and the surrounding, which is growth factors. This, okay, so he puts like tables here, I know in the slide, and I put the same ones just because he, like, I tried to look for other easier ways to memorize, but all of them complicated a lot. So just focus on his slides and I'll tell you what part to focus on. So growth factors, again, from the name, they are factors that help the cell to grow or regenerate or repair and so on. So again, they will help in cell division, proliferation, and they will help in the cell survival in case of any injury, okay? So, okay, these two tables he puts here, I'll tell you what to focus on. So sources, sources mean where did these growth factors affect, right? Which cell? Like here and here and here. You don't need to know too much about them. As far as I know, like he never used to ask us about these. Just know the most important function out of all of them. So there are certain functions that is more important than others. For example, there's formation of like scar tissue. So what growth factor help in the scar formation? Okay. Another thing is blood vessel formation. What growth factors from here help in forming new blood vessels in around the injury? Okay. Another thing is, for example, enhancing just proliferation in general. So most of these cells, I'll go through like in detail now. So most of these growth factors will have the function of proliferation. Okay, so you see here, for example, it will not this one, but there's like stimulates proliferation, enhances proliferation of cells. So in a way, that's like the most common function between all these growth factors. From the name, it will stimulate growth by helping in division and survival. Okay, that's the main thing. If you want to like memorize, if they're like, I can make it easier for you guys to kind of like memorize this. For example, epidermal growth factor will help in epidermis is like the skin, the topmost skin layer, right? So anything that has to do with keratinocytes, which are the skin cells on the epidermis, will be affected or is helped in terms of like proliferation by the epidermal growth factor. So epidermal growth factor from the name epidermis will help keratinocytes, and it will, this part I'll get into this in a minute, but basically it will help form the scar tissue, okay? This is called granulation tissue. So again, wait, I think I can write. So I'll just tell, like, underline things to focus on when you're studying this is keratinocyte with epidermis. He rarely asks about this, like there are other important things he asks about, but just in case. So keratinocytes, and it will also form the scar tissue. I'll just circle this because there are other things later. Now, transforming growth factor alpha, don't forget that you have transforming growth factor alpha, and there's something called transforming growth factor beta. Okay, they have two different functions, and it's very important that you don't like mix them up if you see them in the MCQs and so on. Okay, so wait, let's finish from this slide and then we'll move to that. So this one is alpha, okay? It will stimulate, again, proliferation. You don't need to know specifically for liver. It basically helps in division of a lot of cells. You see hepatocytes and other epithelial cells. Again, he like you don't need to know like the specific cell because it's too much detail. Like I don't think he'll ask this for you, right? Okay, so hepatocyte growth factor is more specific. From the name, you can know that it is for hepatocytes. So if he were to ask you, it would be more of from this name. So hepatocyte growth factor for hepatocytes or liver cells. Again, yeah, this is, you don't need to know, like, for example, these, it's fine. He won't focus too much on that. This one was like, it's one of the most important ones. Why? Because even from the name, you can tell it's a bit different. Like it's vascular endothelial growth factor, right? So all of these before had only growth factor, growth factor, growth factor. This one is vascular endothelial, like it's specified more. So you can memorize this by the word vascular, and it will basically, yeah, it will stimulate the endothelial cells of the blood vessels and increases basically to do with the blood vessels. There's another term for this. I don't know why he removed it for you guys, but there's a term called angiogenesis from the name. Angio is blood vessels, like genesis is making. So making of new blood vessels will fall here. So angiogenesis, also from the name, vascular endothelial growth factor, making new blood vessels, as well as like increasing vascular permeability, all will fall under vascular endothelial growth factor. For short, it's called VEGF. You might have seen it here to there, but this is what it is. So again, let's try to sum up. So epidermal growth factor, from the name epidermis, is for keratinocytes, your skin cells. And there's a additional function, which is for the scar tissue or granulation tissue. I'll also talk about this more, don't worry. Transforming growth factor alpha is basically proliferation. It will help in a lot of cells. Hepatocyte growth factor, specifically more for liver cells and other cells, of course. And this one, vascular endothelial growth factor, is for your blood vessels. So anything to do with blood vessels, making new blood vessels, proliferation of blood vessels, or increasing permeability is all under VEGF, vascular endothelial growth factor. Okay. And wait, let me go to the next one. Yeah, here. So this is the other set of growth factors. The reason why I'm going into like detail for this is because he asked us, I mean, for our batch at least, he focused on this one, fibroblast, which I'll explain, fibroblast growth factor, and again, the one I keep focusing on, this one, VEGF. Okay, so I'm trying to just like go over all of them just in case he asks, do we need to know? No, that's what I told you guys at the beginning about the sources. So sources, not really, he won't ask, because you see how overlapping they are, like platelets here, platelets, macrophages is mostly in all of them, and so on. So no need to focus on the sources. It's more of the function that he likes to ask about. And it's like, that is more important to understand. Okay. But good question. Yeah, I was also confused in second year about this. Okay, so platelet-derived growth factor, from the name platelet. Okay, and this is another important thing because platelets are involved in in the repair process in general, and it will affect fibroblasts, smooth muscle cells, anything you can think of is under platelet-derived growth factor, and you'll see that later. The important thing from this one is that it stimulates the extracellular matrix protein synthesis. Because again, remember we said the matrix is important, so PDGF, platelet-derived growth factor, is important in kind of maintaining the matrix so that the regeneration or repair can happen. Okay, and again, it can stimulate proliferation, division of all these kind of cells, and it can act also as signals for white blood cells. There's like an easier diagram I'll get back to, and I'll talk more about this one. The other thing is fibroblast growth factors. This is, I guess this is like more important because it has a unique function again, which is angiogenesis, okay? And this one, ECM. So angiogenesis, from the name, like I said, it's making of new blood vessels. Now, I know the name is kind of, even it got me confused before when I was studying. Fibroblast is, you'll think that, oh, it's only for fibroblasts, right? It makes you think that, but it's actually, it can work for vascular, so endothelial cells for new blood vessels. And so this one is extracellular matrix protein, which is also from the platelet. Again, the names don't have to like necessarily match the function, but this is how they are. For fibroblasts, of course, like fibroblast is there, which is like easy to memorize, but again, this is unique kind of, that's why he likes to focus on this. The other thing is, yeah, so transforming, we talked about alpha, which is in general, it can stimulate proliferation of liver cells and other types of cells. Beta is important, more important than alpha, and something he could ask more about. Why? Because again, see, it will stimulate the extracellular matrix protein synthesis, okay? And the same thing, so fibroblast, whenever you see fibroblast, it comes most of the time like hand in hand with extracellular matrix, okay? The last thing here is keratinocytes. From the name, it's like the epidermis one, if you want to think about it. Keratinocyte is for keratinocyte, like from the name itself. So you don't need to, like it's not that hard to memorize this one and the epidermis one. Again, like I said, focus on the kind of like the unique functions and things that are in common with each other. For example, he could ask you which one would like help in extracellular matrix protein synthesis, or keeping the matrix, let's say, stable, or help in healing. You could, and the choices could have, for example, this one, and other like wrong answers, but this would be the answer. You get my point? So it could be this one that he put, or this one, like fibroblast. The thing, like, you can think of it like the second set of this table is more for the extracellular matrix. The other one was for more of like proliferation, like division of cells to help in the regeneration, right? I hope that's clear. Okay, so just remember, like I'll try to mark them when I send the new PowerPoint, but this is what I want like you guys to focus on, because this is what he focuses on. Okay. Okay, so I just like to summarize. Yeah, I'll summarize. Yeah, no worries. So epidermal growth factor, again, for your skin cells or keratinocytes. So epidermal growth factor goes hand in hand with keratinocyte growth factor. And you can say that they sort of have the same function. They help in skin cell like regeneration. The unique thing, which is here for epidermal growth factor, it's to make the scar tissue or granulation tissue. Okay, you can memorize like this one came first, like in the table, and this has the unique feature, which is granulation tissue. Keratinocyte is more specific for keratinocytes, so you can think of that as the main function for that. Epidermal has both, okay, granulation and keratinocyte. Now, transforming growth factor alpha is for stimulation proliferation of a lot of different types of cells. He doesn't go, like, you don't need to know in detail, like, for example, these or where they're used in. This is like in general. You just need to know that they are growth factors for a lot of different types of cells. Hepatocyte, from the name, it's more specific. So you can think of it that it will more of help the liver cells like divide and proliferate in case of injury, okay? VEGF, vascular endothelial, also from the name, it will help in the blood vessel formation, angiogenesis, and permeability and proliferation in general for blood vessels. Okay, so blood vessels, liver, many types of cells, skin cells with scar tissue, okay, because it has a different name from keratinocytes. The second table, platelet-derived growth factor, is in a lot of things. You'll see PDGF in a lot of things. So like white blood cells, smooth muscles, and so on. The important thing, again, in terms of tissue repair and so on, is the extracellular matrix formation. So extracellular matrix formation by PDGF, by fibroblast growth factors, and transforming growth factor beta. Now, fibroblast, so we finished this part. Fibroblast growth factors, fibroblast, the matrix, and angiogenesis. That's the third one. So angiogenesis, this one, and the VEGF is for angiogenesis, or making blood vessels. Transforming growth factor beta, you can think of this main function is for the matrix in general, okay? Make the extracellular matrix synthesis. And then keratinocyte for keratinocytes. Does that make sense? Like the three together here is for mainly for extracellular matrix formation, okay? Was that like a bit easier to memorize? I'm sorry, like because it's a lot of memorization mostly, but I tried to make it easier. Okay, good. Now there is a picture for all of this, which is in the second lecture, like tissue repair 2, but I'll bring that again up there. But here, I just wanted to show you guys like what he put. So here, it's a nice summary. For example, this one, oh, sorry. Uh, Wait, yeah, sorry, I couldn't see because of the video thing. Okay, so debridement, these things are not really growth factors. So this is what we'll talk about in the second lecture. If you want a good, like, quick summary of the growth factors we just talked about, you can know from these. See, chemotaxis, the proliferation of fibroblast keratinocytes. Do you see the first one here is platelet-derived growth factor and transforming growth factor beta. In my opinion, these two are very, very important in terms of like extracellular matrix, fibroblasts. Okay, fibroblasts, extracellular matrix come hand in hand, remember? Okay, these others are cytokines. Again, they're not brought up in this lecture a lot, but if he does like talk about it, this stands for tumor necrosis factor, IL-1. This is more of in immunology. But from this lecture, just try to understand the growth factors, okay? Because that's the most important one, these two. Angiogenesis, do you see here? Vascular endothelial growth factor, fibroblast growth factor, especially number 2, okay? So here there's number 1, acidic, number 2, basic, but 2 is more involved in angiogenesis. You can remember it as like vascular endothelial for vascular is number 1. The fibroblast growth factor is number 2, okay? So the second one is important for angiogenesis. And then, okay, PDGF, platelet-derived growth factor, can also help in angiogenesis, which is, I know, like it's contradicting, and this is what we had to appeal in our year, because he asked us about angiogenesis, and none of these, I think, this was there and this was there. And the answer was this one, because again, this was more focused on in the table, okay? So yeah, we appealed it anyway, and like it was fine. But just in case, I don't think that it will happen again. Like, don't worry, it should be like one option that's given. And anyway, if he gives you a choice between this one and any of these two, just choose these from these two, because this is what he focused on, or what was written at least in the table, right? Okay, and then the last thing, see, a remodeling of extracellular matrix, same like proliferation of fibroblasts. You have TGF beta, which is more important, and a platelet-derived growth factor. You can memorize this as by saying platelet-derived growth factor has a lot more function. See in the table, platelet-derived growth factor has many, many more functions than just maintaining ECM. Transforming growth factor beta has main function is ECM. Okay, that's why they put it first. So transforming growth factor beta, more for extracellular matrix, but you also have platelet-derived growth factor. Again, these are just cytokines and other factors, but these two are the growth factors. Okay. Okay, let's recap this again with the table. So these two not related to growth factors. Proliferation of fibroblast keratinocytes. I would put, because they said keratinocytes, you can also include, like when you're studying, just write like here, you can include epidermal growth factor and keratinocyte for keratinocytes. Okay, the two for skin cells. But you also have proliferation of fibroblasts, platelet-derived growth factor, then the transforming growth factor beta. Okay, the same way for extracellular matrix, but opposite order, because the transforming growth factor beta is more of its function is for the matrix than the platelet-derived. Angiogenesis, again, another thing. Vascular endothelial 1, that's the first one. Fibroblast growth factor 2 is for angiogenesis. And PDGF also has slight function in blood vessels. Platelet-derived growth factor, remember, it's like for everything. So you can find it here, you can find it here, you can find it there. Okay, that's the reason. But the important ones, again, the unique ones, are the ones to remember. Okay, is that clear? Is everything clear? I'm sorry, like it's I know it's a lot of things. but I hope I made it easier. Do you have any question, any like concern, if you want me to make something else like easier to memorize? Okay, it's clear, good. Anyway, we have questions on this later, so I hope you like whoever attended was like focusing. It's fine, no worries. But just remember like the tricks I told, like I told you to kind of memorize. It will be easier for you in the exam, like what if you remember them. Okay, so now we'll talk about, yeah, so now we talked about cell types. We said labile, stable, the type of cell determines whether it will undergo regeneration or repair of the cell. We talked about growth factors. We said there are certain growth factors that will help certain cells like in terms of regeneration or repair. Now, there are receptors that you need to receive these growth factors, to read these growth factors as signals to be able to heal itself, right? So these receptors, again, I know it's a table, but there's a picture that can like help simplify all of this. So there's three types of receptors that are like more important, like that are important to know. There are receptors, so I'll go to this one. So this is the picture. Receptors with intrinsic means with its own tyrosine kinase activity. So in receptors, again, it's important that you have the growth factor or cytokine or whatever that comes to it as a ligand, okay? It will come to the receptor. It's important for that receptor to be able to read or process that growth factor, right? So we have one type which has tyrosine kinase within the receptor itself. And tyrosine kinase, whenever you see that, it's to basically, from the word kinase, the enzyme, it adds phosphate to it. So kinase will add phosphate to the signals so that they can go through these pathways, which I'll get into. So again, receptors with its own tyrosine kinase activity, okay? And the second one is G protein. This one is very common, like in a lot of cells, but basically G protein receptors are like transmembrane receptors. So they basically fold a lot of times. And I'm sure you've taken, like, this will come up in a lot of other lectures, but basically it's one of the receptors that can process like a growth factor or anything else, by the way, like not only specific to this. So G protein coupled receptors, we said receptors with its own tyrosine kinase activity and receptors without its own tyrosine kinase activity. So that means without its own means that it will have like other tyrosine kinase factors might help this to process the cytokine or growth factor, okay? I hope that made sense. So this is what the table is talking about. They're saying there are receptors with its own tyrosine kinase, there are without, and there are G protein coupled receptors. Again, ligands, not really important because see, it's very like general, like very broad. You have hormones here, and the growth factors, the main like growth factors, the main receptors that they attach to is this one, okay? With some exception, they will go here. Okay, so again, it depends. But mainly, if you know that most of these attach to the ones with its own activity, tyrosine kinase activity, that's enough. And this I'll try to talk about them with the picture because it's more like clear. So let's talk about the first one, intrinsic tyrosine, like its own tyrosine kinase activity. So when a growth factor comes and attaches here, it will phosphorylate like itself, like its own self, and it will also like bring forward other substances, other kinases, okay? And there are three different pathways it can go through. PI3 kinase pathway, this one, MAP, and IP3. I know it's a bit like it's all over the place. But the main important thing is that for you to know that this is how it processes that growth factor, and that's the way it can go to the receptor and kind of change the cell to divide or tell the cell that it needs to divide. PI3 kinase, so you see all of them or most of them see kinase pathway. If you know that, like I memorized it like this, honestly. So the ones with its own tyrosine kinase activity has the kinase pathway. Okay, so PI3 kinase, MAP kinase. And this one is IP3. I know it's a bit different, but IP3 is the inositol triphosphate pathway. Okay, uh, you can memorize this by saying that G protein is the CAMP pathway, right? So G protein always uses AMP or cyclic AMP, the CAMP pathway. If you know that, then it will be easier to know that IP3 is for the other receptor, which is with its own tyrosine kinase. Okay. I hope that's okay. There's another slide for this one, which I'll also talk about. So kinase pathways all fall under this, with the addition of inositol triphosphate, which is separate from the cyclic AMP, which is always with the G protein coupled receptors. Now, the third one, without its own tyrosine kinase, so if they don't have their own their activity, their own tyrosine kinase activity, what will they do? They will recruit something called JAK-STAT pathway, okay? This is another substance again. This will help it to kind of phosphorylate or make up for the receptor that does not have. So JAK-STAT pathway, you can see that it's a bit different from the rest, like it sounds different from the rest. And it will, so whenever you see like JAK, that's like, it will help phosphorylate, and like the receptor itself with STAT, it will also phosphorylate this. This is like a cell communication lecture, I think, which they'll talk more about in detail. But JAK-STAT pathway basically is for the ones without, to make up for without the kinase activity, okay? And that's how it will all go to the nucleus and tell the cell that it needs to divide. Is the receptor part clear? So there's three types. That's my main point. Growth factors mainly attached to which one?
Tyrosine kinase.
Yeah, the ones with intrinsic tyrosine kinase. Good. So this, okay, so there's another picture that he puts, which is this one. Sorry if it's not, yeah. But so he talks more about the intrinsic tyrosine kinase factor. So I tried to like highlight or like show you what how I studied this, but basically he tells you like you don't need to focus on like how it works inside, like you don't need to know if this activates this, no need. Just know that again, growth factor with the tyrosine kinase receptor it can phosphorylate on its own. See here, it can phosphorylate on its own, and it has kinase pathways, it has phospholipase C pathways and so on. Okay, which is not the focus of the growth factor. But whenever growth factor binds to it, it will form complexes. Again, you don't need to know in detail like these names, I just highlighted to show you. So it will have complexes, and it will have something called RAS, okay? So RAS will be activated, which will activate RAF, and so on. So these are just kind of like downstream signaling pathways. This one, okay, is involved in the MAP kinase cascade. Remember, we said if you remember the word like kinase, that's with the tyrosine kinase. So see here, MAP kinase, and you see the same thing, RAS or RAF. Basically, these are like intracellular signals, okay, for this pathway, for the MAP kinase pathway. As long as you can like remember that, that's enough. Like for me, I never like he never asked about or asks about this, for example, kinase, these different kinases. No, he just wants you to know that the MAP kinase one is under tyrosine kinase receptor using RAS, okay, or RAF, like these two signals. So that's the main thing. That if you can remember that, that's good enough. This one, CPI3, you can memorize, like you can just know that PKB, honestly, it's not as important as this one because they tend to focus more of this because it comes with the growth factors, okay? Whenever a growth factor binds. Is that clear? Is that like, okay, yeah, sure. So, okay, I'll try to talk about like a summary of these three and then go into RAS. So we have receptors on the cell ready to receive the growth factors, cytokines, ligands, anything, okay? The main thing involved with growth factors is receptors with its own, it can function on its own in tyrosine kinase activity, okay? G protein will come with others, in every type of cell there's G protein for other kind of ligands, anything else, and it will have the CAMP pathway that you guys will have another like lecture. This is for, I think, physiology, more of physiology. But G protein, CAMP pathway, okay? Whenever you see G protein, that's CAMP. And the last one, without its own tyrosine kinase activity, something has to make up for it, right? So it will recruit signals called JAK-STAT. So it's a bit different name from the rest. So JAK-STAT for the ones without, to kind of make it like to make up and make it faster to go. So it will like it can take it there to the nucleus, okay? Okay, now we said our main focus is here, intrinsic um, tyrosine kinase activity is the one that receives the growth factor most of the time, as you can see in this table, okay? Now, it has different pathways. It has the most, if you can see, it has 3 different paths. The most important one that they like to ask about is something called MAP kinase pathway, which is the last thing that I talked about. So MAP, any kinase name that you see here falls under the tyrosine kinase receptor. So the MAP kinase, RAS and RAF, like they are intracellular signals that are involved in the MAP kinase pathway. So these are most of the time like they're kinases within the cell. So for example, this receptor, okay, the reason why they showed like this alone is because when there's no receptor, it's like this, okay, it's 2 separate things. But when a growth receptor comes, it fits the shape and 2 of them kind of like join together to telling the receptor that, okay, now I'll phosphorylate myself to recruit all of the other signals. So, okay, so this is the tyrosine kinase receptor with the growth factor. It will phosphorylate itself and recruit other signals such as RAS or RAF, which is again involved in MAP kinase, MAP kinase. So kinase, and RAS or RAF. Okay, these are other kinases downstream. Like you don't need to know the names of it. He didn't mention it too much, but this is like the names are the most important part. Is that clear? Or do you have like specific questions or? Okay, good. So I got an example of a question that they could ask you. Again, this is not, but we can read through it and see. So in an experiment, various soluble mediators, whenever they say mediators, just think of growth factors, are added to epidermal cells. When epidermal growth factor, okay, yeah, when epidermal growth factor is added, it binds to epidermal, okay, including RAS protein activation. See, which of these, like which of the pathways, basically they're asking, involves RAS proteins. That's how they can ask you. Like they want to ask you, what are the kinases downstream? Or they want to ask you like, what does RAS even do? You know, just know what pathway is it involved. And the main function is it takes the whole signal, the downstream signal, to the nucleus so that it can help itself, the cell divide. So, okay. Okay, someone answered, which is good. So yeah, you see how it can be asked. Why is it mitogen activated protein kinase? I know it wasn't mentioned like before, but that basically stands for MAP kinase. Okay. So whenever you see anything for MAP kinase, that is involved with the RAS protein activation. Okay, calcium ion channel, no. Cyclic AMP is with which receptor?
G protein coupled receptor.
Good, yes, G protein. Good job. So G protein is with cyclic AMP or CAM, to memorize it quicker. Then cyclin-dependent kinase, we didn't talk about it here, but this is basically for cell cycle. You don't need to know. You don't need to know this at all. If you didn't take it, just cross it out. Like this one, this one. This one was for G protein. JAK-STAT was for which one?
The one
Yeah,
that doesn't have the intrinsic activity.
good. Yes, good job. So see how it's like asked, like the choices. This is how they can ask you. And the remaining one, MAP kinase, is for the RAS protein. That's what I wanted to kind of like focus on. Like these are proteins that kind of get activated step by step by step, just to make it go to the nucleus, like the whole signal. Okay. Here, yeah, even here, it says the same thing. I'm just making sure I covered everything. Yeah. Okay. Now, so I hope that's clear. Okay, so this I just want to recap like quickly on the things we took. So cell type, we took cell type, we took growth factors, the types, remember the table, and we took the receptors that can intercept these growth factors, okay? And the most important in our case is the tyrosine kinase one. There are different signaling types. So we took growth factor, we took receptor. There are different signaling types between them. Again, it's not the main like focus of this lecture, but since he included it, I just want to go through it quickly. Like there, you have autocrine from the name auto. It's like stimulating itself. So the cell releases like signals, growth factors, cytokine, anything, and it will go to its own receptor to kind of stimulate, like to kind of stimulate or trigger its own cell. Paracrine, from the name para, means next to, so it will stimulate or attach the receptor next to it, next to the cell. Endocrine signaling, from the name, endocrine has to do with like hormones and everything. So these will go and use the bloodstream. So you have hormones in your blood. So just think of it like that. Like the hormones will go through the blood and it will affect cell distant or away from the original cell. Okay. So these are just types of signaling. So auto, paracrine, and endocrine is hormones. So it uses the bloodstream. I hope that's clear, because now we'll move on to the fourth and last, like the final important thing in determining where the cell will go once there's an injury. Will it go to regeneration or repair, right? I keep repeating it just so it gets easier when you study it. So, okay. The last thing is extracellular matrix. Now, extracellular matrix, I want you to think of it as like I said before, support around the cell, blood vessels, anything you can think of. It's the extracellular matrix. There are two components of extracellular matrix. I'm sure, like, I think in histology, you'll take it more in detail. But for now, just know that there is interstitial matrix, whatever is between the cell, whatever lies here. See all of these between the cell, between blood vessels, these are interstitial matrix. And the other thing is basement membrane, okay? Basement membrane is kind of like right below the cells. See, right below the cells, right next to blood vessels. This is all basement membrane. Whatever is in between the basement membrane, between the cells, everything is like interstitial matrix. Now, you can think of it as, you know how a cell has cytoplasm? So the interstitial matrix is like the cytoplasm for our body. Okay, it holds water. It can hold all of these things together. It has proteins, collagen, and fibroblasts. So these are fibroblasts. So it has cells of its own, just like a cytoplasm holds organelles in one cell. Okay, so this is what I mean. You can think of it as kind of like a support, like water, just to hold everything together. So it has collagen, elastin, fibroblasts, which are the cells of the interstitial matrix. And so proteoglycan, they're kind of like the water I talked about. They're called GAGs in histology, like, but basically proteins and sugar together, okay? Basement membrane is the other thing. Basement membrane, like I said, it's kind of like an immediate support to the cells, okay? So it lies beneath the cells. surrounding blood vessels, and so on. It is made of, this is important to know because he likes to ask what type, but it's made up of type 4 collagen. Type 4 collagen is specific for basement membrane, like all of the time. And it can have laminin, proteoglycan, just for extra support, you know, for proteins. So this is what I tried to talk about. Matrix is the network that surrounds cell, okay? Whether they're like the interstitial matrix, whether they're like the water for the cells, or whether they're basement membrane, whether they're holding the cells, epithelial cells, blood vessels, and so on. The interstitial matrix is the space between cells. It is made by fibroblasts or mesenchymal cells. So it is made by these cells. They just keep making and making the more support you need. Okay, it makes collagen, it can make elastin, and so on, and it can hold water. These are the major constituents, everything that was mentioned before, collagen and so on. Now, basement membrane, again, see, it's arranged around endothelial cells, and like right under the epithelial cells and even smooth muscle cells. And this basement membrane is made by, again, type 4 collagen. Which cells make it? We said for interstitial matrix, we said fibroblasts make it because fibroblasts are the constituents of the interstitial matrix. What makes the basement membrane, which is between the cells and the spaces, are both of them. So what I mean by both of them is they're made up by the cells above the epithelial cells, and they're made by the cells below them, which is the mesenchymal cells or fibroblasts and so on. See, it says here, it lies beneath epithelium, around blood vessels, around smooth muscles. It's synthesized by the epithelium above and the mesenchymal cells below it. So it's kind of made like by both up and down, okay? So that's why, and it says, he says, they kind of form like a plate-like mesh to kind of support the cells, okay? The main important thing is that it's made up of type 4 collagen and laminin and other proteoglycans. Is that clear for everyone? So we talked about the matrix in general. There's two components. Matrix, the space everywhere within the matrix, and basement membrane, which, like, you can think of it, it's outlining everything else. It's outlining the cell, it's outlining this one, the blood vessel, smooth muscles. Okay? Is everything clear so far? Like, do you have any questions? Because this is the last slide. Like, I'll just briefly talk about functions, which we already talked about. So basically, functions of extracellular matrix, it's very easy to understand once you know, see it, like if you imagine that it's like supporting everything, right? So it supports the cells, the tissues, and everything. It will, and that's the same point for this one. It acts as a substrate for cell growth, means that it kind of helps the cell when it regenerates or divides. It will hold the shape, okay, for around the injury. And it regulates, again, these 3 points, same thing. As long as you understand that extracellular matrix is supporting and it will regulate the division of cells, that's enough. Like, that's good to know. Which brings me to the next point, which is intact extracellular matrix is required. Like, you need it for regeneration. Remember the first slide, we said regeneration or repair. Extracellular matrix will determine, like, if there is good extracellular matrix around the cells, if there's any injury, it can go back to normal just because it has a stable extracellular matrix. If the extracellular matrix is damaged, repair can be done, which is the second path with scar formation. Okay. Now, we talked, so there are some questions based on the first lecture, this lecture. I just want to sum up everything by asking you guys. So for example, we said there's two pathways, regeneration and repair. Regeneration is back to normal, repair is partially back to normal. So we said 4 things. So what type of cells will make regeneration possible more than repair?
Stable and liable.
Good, yes. So these two types will make regeneration possible. Permanent will not. Okay, growth factors, I can't ask you a lot about that, but basically if you have growth factors, that will make regeneration possible. If you have receptors with tyrosine kinase, it will make regeneration possible. What about extracellular matrix? We need that for regeneration. Okay, so that's what I wanted to focus on for the first one, because he talks about that. Now, there are a few questions, like I think 2 for this lecture, like we can answer them together. You can type it in the chat. So 50-year-old woman tests positive for hepatitis A antibody. The serum, so these are liver enzymes, AST are high. 1 month later, these enzyme levels have returned to normal. Which phase of the cell cycle best describes the hepatocytes 1 month after infection? What about the rest? What do you guys think? We'll discuss it anyway, so it's fine. Like whatever you think is the answer. So this is talking about cell cycle. Yeah, since stress is removed. Okay, good. Yes. So the answer is A. Good job. We said now try to understand like what is the question asking you? It's asking you the liver cells, so hepatocytes, and it's talking about injury and which stage of the cell cycle is the liver cell in. So if it was still injured by, oh, by the way, liver enzymes being high, hepatitis A, hepatitis, like inflammation, like there's an injury happening. When that happens, the liver cells, remember, it leaves the G0, it goes and divides and makes up for the damage, okay? Basically, it will help in regeneration because these are stable cells, okay? That's the main point. Now, 1 month after, the liver enzymes have returned to normal, which means the damage is done. As you said, since stress is removed, like regeneration happened, regeneration happened. Now it's back to stable again, back to stable cells. So this question is just asking which cell cycle is the liver cell in, usually, okay, basically. So G0 is the state of the liver cells, state of kidney cells, pancreas, anything under stable cells, okay? Yeah, you can say it like send it private, it's fine, like I don't mind. Okay. Is that
permanent cell,
yeah?
excuse me, permanent cell also in G0, right?
Which one?
Permanent.
Permanent? Oh yeah, okay, I see. Yeah, it is in G0, but it's not able to move out of G0. Does that make sense?
Yes.
Okay, good. Okay.
During the injury, the liver cell was in G1, right?
Yes, so it depends. It can undergo all of these. Like it will go back to G1, then S, G2, mitosis, G1, basically in all of the cell cycles. Do you
طيب
get
how
that? Yeah.
how do we know? Like, are we gonna get tested on which phase they're gonna be during the cell injury?
No. They can't ask you because like that will be for cell cycle like lecture. The whole point of this is they just want you to understand what cell type is liver cells. So yeah, you can basically cross out these because that's not the focus of the lecture, right? Does that make sense?
Yes.
Okay, good. Yeah, they won't ask you ever, but this is how the questions can come with the cell cycles. Okay, this is the second one for this lecture. In an experiment, surgical incisions are made on rats. Observation about the wounds are recorded over a 2-week period using various chemical mediators. Which one of the following steps is most likely affected by transforming growth factor beta? And it has options, leukocyte extravasation, increase in vascular permeability, production of collagen, chemotaxis of lymphocytes, migration of epithelial cells. Even if you see overlapping functions, just choose the main one or its main function for transforming growth factor beta. Okay, yeah, I got one answer. Like, what do the rest think? Which is very good. Okay. Okay, so good. So most of you said C, which is the correct answer, and I know like there's a certain confusion. So someone said E and someone said B. Okay. Yeah, okay, so someone is saying that the questions are from Robin. I was going to say, yeah, Robin, mashallah, that's really, really good, by the way. Robin's is very, very good, and this is where like the doctors like to get the questions from, especially Dr. Oban. So I recommend it really. All my questions are from there anyway. It's good practice either way. So I'll try to explain why the answer is C. Okay, as someone correctly said, TGF-beta will mostly affect collagen production. Now, I didn't say, the slides didn't mention collagen, but collagen is the one involved in what? Extracellular matrix. We said, wait, let's go back to the slide really quick. We said, see, oh wait, this is okay. This is like a summary. Transforming growth factor beta mostly involved in extracellular matrix protein synthesis. Extracellular matrix, we mentioned that one of the components is collagen, elastin, fibroblasts, everything that makes up extracellular matrix. That's why the answer is collagen, because that's in the extracellular matrix, and its collagen is used for repair, like it's used like for scar formation and so on, right? Okay, let's see. Someone said B, increase in vascular permeability. Which one is that? And you guys can all answer now, it's fine, whoever knows. Increase in vascular permeability, we said there's two main. Yeah.
It's the vascular endothelial growth factor
Yeah,
and fibroblast growth factor.
good. Yeah, good job. So
Okay.
fibroblast growth factor and vascular, VEGF. So that's why transforming growth factor beta doesn't involve blood vessels. So we can cross that out. We can cross even white blood cells. Chemotaxis of lymphocytes is kind of the same thing as A, because you're removing the white blood cells, right? So you can also cross that out, not the main function. Migration of epithelial cells, also the same thing, like anything to do with migration, moving of WBCs are mainly which growth factor do you guys remember?
Platelets growth factor.
Good, yeah. Platelet-derived growth factor has most of these functions, which is white blood cell, chemotaxis, migration. If you see on the table, transforming growth factor beta doesn't have like migration. Okay, it has proliferation of cells. Okay. Oh, no worries. Yeah, we're all Yeah, whoever answered B, like, don't worry about it. As long as like you understand now, that's good. Is it clear, the two questions? So this is how they can kind of ask you like growth factors and so on. Okay. So this is the second lecture, and I think it will take shorter, but do you all want a break, or is it okay to continue? Okay, yeah, sure. We can have like 5 minutes break if that's okay, or 10 minutes. Like this is the bulk of the second lecture, which we'll talk more about healing and how wounds heal in terms of repair. Because regeneration, we said it will go back to normal, right? Repair is the one where we want to see like how does it happen in terms of granulation tissues, scar formation, and so on. So there's the first two like slides are a summary of the first lecture. So I won't go through like it's basically a summary, like don't worry too much. So it depends, again, the healing depends on cell type, amount of damage, and the support, right? And the same thing, so cell type, if it's labile or stable, it will undergo regeneration. That's all you need to know. This is, by the way, all the text is from his slide, but as long as that's clear, like don't worry too much about it. So labile, stable, and he gave you some examples of kidney and liver. This is like regeneration happens for these two. Why? Because they have stem cells, and there are in case of stable tissue, even if a part of it is cut, there are remaining stem cells, remaining cells that can kind of make up for the injury. Okay. The second thing is even if the cell type is like stable and it can go back to regeneration, if the damage is too much, let's say you take like a lot, like the damage maybe occurred in most of the liver lobe or something, or it was a very severe form of inflammation or anything, it will undergo repair. So just remember that there's also the injury. It depends on the amount of injury. If it's too much, it will undergo repair. Again, repair, whenever you hear the term repair in this lecture, it's about fibrosis or forming a scar. And that will have 4 processes like that will make the scar. And granulation tissue is also, they also come in hand in hand, but basically it's a new type of tissue which will lead to scar formation and so on. So okay, so scarring, the main like objective or main how do you say, the result of scarring in repair is to make granulation tissue. Like I said, it's a new type of tissue to make up for the injured space. So new type of tissue, granulation tissue, and to turn this into collagen or fibrosis, and remodel that kind of tissue to be able to live or be within the original tissue. Do you get my point? Like there is a new tissue being formed called granulation tissue, and it will turn over time into like fibrosis. It will undergo fibrosis through collagen and so on. It will be remodeled to kind of be able to just survive in this normal cell. This is scarring. Like this is what happens in repair. There are 4 things that happen. So you need, so for to make new tissue, you need what? You need nutrients, you need supply, right? What is that? You need blood supply. So that's the first thing. New vessels formation, again, angiogenesis is needed to make the new granulation tissue or the scar tissue. So new vessels formation is one. The other thing you need is to make the new tissue, you need collagen, right? So you need fibroblasts to make the granulation tissue. And same thing, like if you see fibroblast proliferation, that means collagen will come in and form the scar from the fibroblasts. And then the last thing is remodeling of scar. That falls with turning into, or like remodeling it after the granulation tissue is formed. Okay, so 4 components. You have blood vessels. You need new blood vessels to make the new tissue, granulation tissue. So new blood vessels. You need collagen which is made by fibroblasts, and remodeling. Oh, okay. I missed. Yeah. So okay. So this is by the way what we're starting. I just recapped the last lecture, and I'll recap scarring for you. So scarring, we said cells can undergo scarring if they're in the repair pathway. Okay. This is when the damage is either too severe, or if the cell is not able to divide, or if there is no not intact extracellular matrix. Now, the summary of scarring is to make a new tissue. So imagine there's an injury, like a hole here. You have to cover it with new tissue, and you have to kind of it will be remodeled over time. Like your skin will not always look like that if you have an injury. So this is the granulation tissue. Over time, it will be better. Like it will look more like the surrounding area, right? It will not always be open like this. So this is what we call remodeling, okay? So that's the objective of scarring. So scarring in repair, making this red thing, the tissue, and being remodeled over time to be more in sync or like in line with the surrounding area. 4 things that will affect or that will lead to scarring is formation of new blood vessels. You need blood vessels, you need collagen, and that's why you have fibroblast and collagen. But yeah, they're separated into 4. But fibroblast and collagen for the scar, and you need to remodel. Like I said, you need to, like, over time it will be stronger and a stronger scar, let's say, okay, to cover the wound. Is that clear? Like this is what I started. Like this is just what I said. Okay. Okay. Good. Now I'll go into like details. So this is the timeline, like the general timeline for the scar formation, and granulation tissue is within that. Like just don't try to confuse granulation tissue with the end result of scar process. Okay, granulation tissue is in the middle of that process. So okay, let's say you have an injury. Within the first day, 24 hours, what happens is like the cells, endothelial cells, endothelials for what? For blood vessels. So blood vessels will start to form, fibroblasts will come, and this is what happens. Like they will proliferate and they will come to the site of injury. Over the next 3 to 5 days, you will have the new tissue formed, which is granulation tissue, from these components. From these components, you will have, see, new blood vessels, new collagen. So collagen was made from the fibroblasts, and now you have a new tissue in place called granulation tissue. Granulation tissue essentially is like tissue with new blood vessels and new collagen to kind of stabilize that injured area. Okay, so see, granulation tissue, 3 to 5 days. Now the timing is important because they can ask you, like for example, so this, I take it as 3 to 5 days as, let's say 1 week, so 7 days. There's another slide where he says 7, so that's why. So 1 week around, so 1 day is the cells start to come recruited and divide. 1 week is granulation tissue, the new tissue is formed. And then after weeks later, which is like 1 month, maybe, you can consider that it will be remodeled. See, the granulation, the new tissue is gone. Now it is better, like it looks better, and it can like stay within the normal cell, okay, normal tissue. So now you have remodeled scar and dense fibrosis. So the scar is stronger with time. So I used to think of it like 1 day, 1 week, 1 month to make it easier to memorize. But yeah, this is the timeline. Okay, all good? I hope it is. If you have any questions, just let me know. I'll touch back again because he has a slide. Okay. Now that's the timeline we talked about. Now angiogenesis is very important to know. Again, which growth factor you already know, VEGF, right? Vascular endothelial, and the other one is FGF, fibroblast 2, fibroblast growth factor 2. How does angiogenesis happen? There are 2 ways it can happen. So vascular and like the growth factors we talked about can happen from existing vessels. So that's the second one. So this vessel is already there. It can kind of grow out of that existing cell using the growth factor, using the signals to kind of make new blood vessels. Okay. The other thing, you can also have endothelial precursor cells. So precursor, if you see that, you can know that it comes from somewhere else, which is bone marrow, precursor, so before the blood. So bone marrow has these cells which can go to the blood and kind of trigger it to make new blood vessels. That's the only difference, okay, between endothelial precursor and pre-existing using the growth factors. So that's angiogenesis. Okay, what else? Now, this is in the first 24 hours of injury. Again, do you see here major involved is VEGF, vascular endothelial growth factor. In which type of angiogenesis? In the second one, in the pre-existing vessel. This one you usually you're not asked about it, but it's good to know because like it comes from the bone marrow and stimulates. So again, this is existing vessel. Let's say there is an injury. You have this growth factor released, angiogenic factors. This will help kind of the new blood vessels to spout or kind of come off from it by stimulating the endothelial as well as the basement, like the membrane here, to separate and to grow from there. Okay. And yeah, this is mainly it. It will proliferate. There's another picture, and then remodel into new blood vessels. So it went from here, this part. It stimulated this endothelial cell. Basement membrane like is here, and there is another cell called pericyte. It's like next to the basement membrane, but it's basically another cell over the basement membrane, they will all form like in the new blood vessel. So the new blood vessels comes from the endothelial cell, like this one inside, okay, in the inside cell, this one became green. And then it will form, the rest will just follow. It will proliferate on top of the new cells as they're formed, and it will result in that. The main important thing again to know the growth factor and how does it do that. Like it will stimulate the endothelial cell to kind of okay, endothelial cell to make new blood vessels, as well as the pericyte from separating from each other in the beginning. So once they separate, this is able to grow, right? So proliferation, endothelial cell, and then remodeling to like new blood vessels. I hope that's clear for angiogenesis. Again, this is within the same like day of injury. This is what happens. Now we'll move on to what happens later. So 24 to 72 hours, again, around that range. So 1 day to the 3-day mark, which is like timeline is here, like from this to this part, is angiogenesis, recruiting fibroblasts and so on. So new blood vessels make sense now, and proliferation of fibroblasts also makes sense. Now the vessels, okay, this is it's the same thing. Vessels are basically more leaky because you're making new blood vessels. They're not as strong as the ones originally, like the pre-existing ones. They're still weak, so everything's kind of like watery. Like there's lots of fluid. That's why they're saying edematous. Like there's a lot of edema. But again, the main thing is these two. Okay. And the new blood vessels are not as strong as the old ones. That's why. Now, 5 to 7 days. See, so he likes to like he might ask like 1 week, and that's the same thing as 7 days. So 5 to 7 days or 3 to 7 days, you have the new tissue formed, which is granulation tissue. I put in like a line here just to show you that this is the granulation tissue with the new blood vessels, with the new forming like collagen. Okay. New vascularization means new blood vessels, and this one will take in a minute, secondary union. But this is basically the granulation tissue. This one, if you see a lot of like blue, like strongly stained collagen, this looks to me more like an advanced, not advanced, but 1 month time, in 1 month time. So I know he didn't put that, but when I checked Robin's, like this is one of the questions had this, and it was 1 month later, you know. So this is more of the remodeled granulation tissue. Okay, in the timeline, granulation tissue is in the middle. Then over time, it might go into that. It might turn to look like this with thick collagen. See, the blood vessels are not as leaky anymore, so they're made better now with the cell. Okay, I hope that's clear. This is all about granulation tissue, and the important thing is you know the timeline and you know what granulation tissue consists of. Okay, is everything clear? We have healing left, and I think as if you understood the granulation tissue part, like it's all good, inshallah. Do you guys have any questions, or is it okay? I hope it's okay. If you have any questions, just like stop me anytime if I was too fast or anything. So now skin wound healing. I tried to summarize like in a few like slides because there's basically 2 types of skin wound healing. So now we're talking about how, so we talked about wound healing in general, like in repair. Now we're talking about the types or how it can undergo that, right? So there is something called first intention or primary union from the name first and primary, and there's secondary intention or second intention, secondary union. Okay, what's the difference between these two is so first, I like to think of first or primary as the first, like the basic way a cell will undergo repair, which is what we just took. So it will have granulation tissue, it will heal on its like by the body healing itself. Do you get, if you get my point? Like the process we talked about, fibroblast, angiogenesis, granulation tissue is all done by our body cells. So this is what primary union means. It will undergo repair by just letting the body heal itself. That's a better way. So it occurs in small wounds that can close easily, and there is epithelial being regenerated. Again, the wording. So like, be careful. So here, regeneration means like in the repair state, regeneration is the proliferation of cells, takes over more over fibrosis, and healing is fast with minimal scarring. So minimal scarring is still scarring. So it's all of this is under repair. It's just that epithelial is proliferation is more than the fibrosis. Okay, why? Because we said less scar, so less fibrosis, more of division and going back to like more normal state than secondary, which I'll say now. So example of this, small cuts like paper cuts are like a classic example. Like any small cut, any small injury will heal by itself and with less scarring. Secondary union or second intention, the opposite. It will have a larger wound, with more fibrosis, healing is slower, and there will be more scarring. Like it will be more obvious that there was an injury. Okay. There's also a term called wound contraction, only happens in secondary. If he asks you, wound contraction is secondary healing or secondary intention. And I'll mention that. Why is that? It's because there is more scar, like more scar tissue, more contraction from the fibroblast and collagen and so on. Now, this
Can I ask you a question?
yeah, sure.
In the primary union, it's regeneration, or it's like part of the repair mechanism that is gonna end up with the scar.
Yeah,
'Cause-
so that's what I meant. So it's repair. Like all of this, now we're talking about repair. They say regeneration as like he said regeneration in terms of epithelial proliferating, like dividing to like this one is not the
So
same
it's
as
not part of like the stem cells regenerating and just covering it.
no. It's part of repair. Yeah, you're right.
Okay.
Okay.
Thank you.
No worries. Okay, so we have primary again, secondary union. Okay, we an example of secondary union, any big injury, any big sort of like burns, let's say ulcer that you see is healed by secondary intention. What that means is it takes time for the body to heal itself, and there's with more scar, like with more obvious like deformity. Okay, infarction is one more like another example. So heart attacks are big injuries compared to like paper cuts. Okay, this is but yeah.
Can
Sorry.
I ask you one last question?
Yeah.
So what is
Yeah.
examples of like the repair mechanism that regeneration that is not part of the primary and secondary? So didn't we say there is a repair mechanism that happens with just stem cells that are regenerating, and then we're gonna have growth factors and basically the types of cells. So what are examples of like things that are basically that's gonna regenerate without any scar?
Oh, wait, good. Yeah, that's a good question. So regeneration are things like that so you can think of it as anything that is continuously happening. One good example is hair cells, skin cells. Remember the example like before in the first lecture I gave you? Yeah,
So it's not really like part of like an injury. You're not injuring yourself. It's just like a everyday life.
Exactly, yeah. It's just like dead skin cells, dead hair cells, So there is no big injury compared to like the repair thing or like this healing. Does that make sense?
Yes, thank you.
Yeah, so that's the difference. Like as long as you can realize that in the exam, that's good. Now, okay, so these are again just like some examples, like he's trying to show you the difference. See here, first intention primary union is you will see like the new like tissue being kind of formed with time, okay? And you have like granulation tissue and it heals within a week, like it's not that much, damage here. And you see like here even it's like you can correlate that to this one. In one day you have like the cells coming, recruiting, angiogenesis, Then granulation tissue is here in 1 week. And then in the weeks to come, you have fibrous. So this is what we just explained, right? So you can see that there isn't much, like, there is a new kind of fibrous region, but not as much as secondary, not as much as big burns and so on. Healing by second intention or secondary union, you'll see a bigger scar, bigger injury, more cells. Healing is very, very slow. You have more like blood vessels and a larger area of fibrosis with what? With wound contraction. That's the other difference. So large area of fibrosis with the small arrows are showing you wound contraction. One other thing I want to mention, secondary union, by the way, because the area of, let's say, the burn is very big, you sometimes need to help the body heal itself. What do I mean by that? It's that, like, for example, this is a skin graft. So for burns or any like other type of injury, the surgeons can put like a new set of like skin on top so that the area is not exposed much, okay? So once they put like a covering, once they stitch, like suture it together, this will help the body heal itself, but again, with a longer, like larger injury, longer time, so on. Do you get the difference? That's why I said primary union, you let the body heal by itself, like completely. Yeah, I will repeat. Yeah, no worries. The primary union, you let the wound heal itself completely. It's a small injury. That's what I meant. So secondary, you can sometimes help the body heal itself by doing skin grafts or like covering the skin, gauze, like with that. Okay, so to help the body be faster in repair. Okay. So wound contraction, I'm gonna explain it now, right now, actually. That's why I just mentioned it just to give you a quick idea that it's only in secondary union. Now, this is wound contraction. So we said in secondary union, it's large surface wounds. So you can correlate that, right? So large surface wounds, secondary union has wound contraction. Now, what is wound contraction? Basically, wound contraction is from the name contraction of cells within the wound. So you have in secondary union, you have a lot of scar tissue. You have lots of fibroblasts. Some of the fibroblasts, they are specialized now because the injury is too big. Like you need another way to kind of close or approximate that wound. Okay, you need to try to make it better to heal. How is that? The fibroblasts can turn into something called myofibroblasts. From the name myo, sounds like muscle, so muscle fibroblast. It has the ability, it's the same as fibroblast, like any, but it has the ability to contract, okay? So if imagine like all these fibroblasts, this scar tissue is here, and within it there are myofibroblasts that will act to like close the wound like that from inside, okay? Does that make sense? That is wound contraction, to try to bring the edges of the wound together as much as possible. Is that clear? Okay, so this is why this is done by, again, myofibroblast. It's a special type of fibroblast. See, it helps to close the wound by decreasing the gap between the edges, okay, of the wound. So that way it's not exposed to bacteria, and it will help it, at least it will help it heal better. Okay, even if it's worse than primary, at least it's better, you know. Now this like this picture is important in another way. It's because it's showing you the growth factors. So we said transforming growth factor beta is important in making collagen and stimulating fibroblasts. This is how it stimulates proliferation of fibroblasts as well as changing it to myofibroblasts to help with the wound contracture, only in secondary union, as well as PDGF. So both of these help in the extracellular matrix, collagen, as well as like changing the fibroblasts to myo in secondary union or second intention healing. Okay, does that make sense? Like you can, this is like a summary. It's better than just explaining. Like you can see it here, like it stimulates that. There is another, oh, sorry, there is, yeah. So this is where he actually uses the picture, like in the second. This is where this one came. Again, it produces large amount of type 1 collagen. Now they're being more specific in terms of wound contracture. The collagen that's produced is type 1 collagen. And so this is in general for the scarring, myofibroblasts for wound contraction. And these are stimulated by PDGF, transforming growth factor beta, and one more thing that was on the slide, fibroblast growth factor 2. So it's important to remember these three, okay? That's why I bolded them. And if I were you, like when I'm memorizing, let's say these ones, I would add another like arrow here and say myofibroblasts for wound contraction with these three. That's why I tried to put them together. So you already know for extracellular matrix is these two are important. And fibroblasts, from the name fibroblast growth factor, will stimulate fibroblasts to go into myofibroblasts. Okay? Is that clear? That's about wound contraction in secondary. I hope that was clear. So this is the most, like, this is important. And the way, like, if you understand this one, that's also good. What else? Okay, we're almost done. So we said that we talked about types of healing. We talked about how wound contraction is within secondary. The last thing, and we talked about the process itself, like granulation tissue. The last thing, the one that happens at 1 month, like weeks to months after the injury, is remodeling, which I said briefly in the beginning. Remodeling means you replace slowly with time, you replace the granulation tissue with scar. This is the final, like the last step. So why do you do that? It's because you can't leave the wound open with the granulation tissue. You need to actually like cover it and be the same or at least better with the surrounding cells. The important thing here is what are the enzymes that help to replace the granulation tissue are something called matrix metalloproteinases, MMPs. So these are from the name enzymes that break down the granulation tissue and replace it with a stronger scar or collagen on top. So collagen, so they're produced by fibroblasts, all of these different type of cells. Okay, so again, the same thing, induced by growth factors, fibroblast growth factor, platelet-derived growth factor. But it's inhibited, this is the only difference between the granulation tissue and this one, it's inhibited by transforming growth factor beta. Now, transforming growth factor beta, the main function, again, just remember, it's to make the ECM, like it's to make the extracellular matrix. But for remodeling, that's why it inhibits the remodeling. Like it doesn't want to let go of the like the collagen it produced. Like it just wants to keep making without breaking it. So that's why it's inhibited, if you want to think about it like that. So it's inhibited by this, but it's stimulated by other growth factors because, again, they have other functions such as stimulating fibroblasts for this enzyme to break the old tissue and make new tissue, okay, the stronger tissue. So this is how it happens. You break the granulation tissue to make new collagen with more stiffness, with more cross-linking and so on. Okay, is that clear for remodeling? Okay, so if you don't have any questions, okay, good. We have this part is, I thought it was not important when I was studying for it, but it's very, very important to understand that wound strength. So at suture removal means like at the beginning of like the wound healing step is 10%, like the strength of the wound is 10%. It will rapidly increase in strength over the next 4 weeks, over the month, and so on. At the 3rd month, again, this is just an example, but over a long period of time, you will have 70 to 80%. What's important here is that this is the maximum wound strength after injury. Like it will never go back to 100% because we said this is repair. It doesn't go back to original, okay? It doesn't go back to 100%. That's the main idea. So 70, 80 is like the maximum it can go back to. after a good number of like weeks to months after the injury. Okay, that's the main point that he wanted to kind of explain here. Okay, last thing, last concept is wound healing, like factors that affect wound healing, what makes the wound go bad, and so on. So the important thing is you have the cell, you have blood supply, nerve supply, fibroblast supply to kind of help in wound healing. That's the first thing. Important is the cell around the cell, important. Okay, you need good nutrition along with blood supply. Like I said, you need nutrition, you need these things. If someone is deficient in protein, vitamin C, they cannot form collagen that much, right? This is collagen diseases. So that's why deficiency in all of these will reduce the effectiveness of wound healing. Other things such as foreign body movement, infection, these things can also affect wound healing in a negative way. And he says the bold ones you see here are in his kind of like he made another slide for them just to emphasize on it. So cell type is important, like we said. Infection, if there is infection, if the person is already weak, immunocompromised, any other disorder, then it will affect wound healing. It will not be like that good of a wound healing as compared to someone who is healthy. Again, diabetes mellitus is under health conditions, so you can say that someone with diabetes has less chances or less like proper wound healing. Okay, and you can imagine diabetic patients, like their foot, for example, it takes time, a lot of time to heal. They have a lot of ulcers and so on. Okay, steroid, I put here how, because, okay, so if there's a lot of steroid hormones, if someone is taking, let's say, steroid hormones, how do you think it affects the wound healing? In a positive or negative way?
Negative way.
Yeah, good, negative way. Why is that? Does anyone have any idea? If not, it's okay, like, let's see. It inhibits, yeah?
It inhibits fibroblast formation, I guess. That's what I remember.
Okay, good. Yes, good. That's it. And someone said it inhibits MMPs. It does that. I think, but the main function is that it inhibits collagen synthesis in general. So even if it doesn't inhibit MMPs, it will inhibit collagen synthesis, and it will reduce the ability of wound healing. Okay, so steroids in general weaken wound healing by decreasing the collagen synthesis or inhibiting collagen synthesis. And yes, even MMPs, yeah, because it does make the new collagen, stronger collagen. Now, what can go wrong if you have any of these bold factors, if you have deficiency, nutrients, all of these, it can have abnormal scar formation, okay? Oh, actually, wait, these are less, so less scar formation, like not enough. These are aberrant, means abnormally growth. So you can have hypertrophic scars, which means from the name, more like expand, like too much scars, let's say. Keloid scars are an exaggerated version of this part. So hypertrophic scar is excess production of scar tissue, and the type 1 collagen is much, much more, and you can see it. Like it's supposed to be like healed, okay, with repair, but you can see a very like distinct scar. So that's not good also. So having too much is also not good. Hypertrophic scar will look like this. Remember type 1, okay, it's like it has excess of type 1. Why? Because keloid scars are excess of type 3 collagen, which is the stronger remodeled collagen, okay? And you see a more distinct like deformity. So keloid formation, keloid scars they have a genetic component also. That's different from this. So keloid scars are more common in African Americans. They are due to excess scar tissue that goes out of the wound. So that's the two differences that you need to know between these two. There's a picture for that. It looks like that. Like it looks, it goes way out of the wound, right? And it's more bigger, and you can see it more clearly than this one. So that's first difference. This is within the wound kind of region. This is outside, goes outside the wound. And the second one is type of collagen, type 1. This is type 3. and this one is genetical. So three differences. This is more genetic predisposition. The third abnormality is something called proud flesh. Okay, so you can see these two were scars, like for sure, like they were scar, like type 1 collagen, type 3. This one is still granulation tissue. So this, we're stuck in this middle phase. We didn't even form the scar, and that's why you can see like it's more pink. It's like more flesh kind of looking, right? And if you like see under that, you will see more granulation tissue more than the collagen. Like you will see more new blood vessels, fibroblasts. So this is called proud flesh, and it's also abnormal. You have more of the granulation tissue. These two are more of the scar formation, which is the afterwards, which comes after granulation. Oh, is that all clear? That's it. But I hope that was clear. You have any questions before we move on to questions? There's three on here, then that's it. I hope that was clear. Inshallah, like this helped you.
Yes, I have a
Yeah.
question.
Yeah.
So the difference between the colloid and the hypertrophy is that the colloid is African American type 3 and it's genetics, right? And that's it. What's the third?
Yeah. Oh, what's the third? I said it's, you see how this one is within the wound region that is
This is one
localized.
outside.
Yeah, that's this one goes outside the wound. Yeah.
Okay. Thank you.
Good job. The other differences are correct. Hypertrophic and keloid scars do not have a granulation. No, yeah. So the granulation tissue is fine. Like there's nothing abnormal with granulation tissue. It's when they reach the third phase, which is the scar formation, that they produce too much scarring. Do you get the idea? Like this one is just more of granulation tissue than the scar. So they're stuck in different like phases, you can say. Okay, yeah. That's the difference between, and just know the pictures because he might but it's very easy to understand, like once you know what's happening. These are the differences. Okay, and okay, so these are like the questions. This was how the picture was in Robbins, but okay, in an experiment, glass beads are embolized into the coronary arteries of rats. This part is not as important, but basically they're studying the heart cell under light microscope. Microscopic appearance is shown here. Which of the following mediators is most likely being expressed to produce this? First of all, like, what is this? And what growth factor is mostly involved? Okay, good. Yeah, someone answered, which is good. Okay. Good job. Yes. So what tissue is this, first of all? You guys can answer.
So basically this is in part of the wound healing, which is basically where
Yeah.
we're producing angiogenesis. So
Good. Yes.
it's E, vascular endothelial
Good.
growth factor.
Yeah, and that's what you all answered. Very good job. Vascular endothelial growth, you can see that angiogenesis, you can see the blood vessels here. This tissue is basically, once you see it, you'll know that this is in healing. It's the granulation tissue. Okay?
Mhm.
All clear? I hope it is. This is the second one. An 18-year-old man lacerated his left hand and required sutures. Sutures were removed 1 week later. Wound healing continued, but the site became disfigured by a raised nodular scar that developed over the next 2 months. Which of the following terms best describes the process that occurred during this 2-month period? From the choices, of course. Okay, good. Yeah, someone answered. What do the rest think? Okay, so good. Most of you said D, keloid formation, which is correct. And I want to say, like, I know the terms here are also, and someone said secondary union. It's fine. Like, don't worry about it. Secondary union does happen, but it wouldn't be like secondary union is a normal process. There wouldn't be a exaggerated scar. It would be a normal like scar, even though it's a big wound, it would be normal scar without having like raised out of the wound scar. Like that, this is abnormal. Prominent, being disfigured, okay, prominent nodular scar outside of the wound. More of shows keloid formation, which we just talked about. So do you see the difference? Someone asked like this, now we're talking about scar. It's fine. It went through the granulation phase, right? It continued, and then when the scar came, it was abnormal. So now we will say that it's keloid formation. That's not like the keloid formation occurred here. Okay. Is that clear for everyone? Is it clear now? Like why it's D? Okay, good. Good job, also everyone. Okay, so a cesarean section is performed on 20-year-old woman to deliver a term infant, and the lower abdominal incision is sutured. Sutures are removed 1 week later. Which of the following describes the wound site at the time of suture removal? So A, granulation tissue is still present. B, collagen degradation exceeds synthesis. C, wound strength is 80% of normal tissue. D, type 4 collagen predominates. E, no more wound strength will be gained after this. Okay, good. Someone answered. I'm happy. But what do you guys like? Just tell me. Yeah. Okay, good. Okay, good job. Yeah, everyone answered the correct, which I'm happy about. So granulation tissue is still present. Now, basic question, the basic idea is they're asking you what is in the 1 week time period, right? Remember, 7 days falls under 1 week. So 1 week, just know it's the second kind of like the time. Second phase is granulation tissue is still present, right? Even like if it's sutured or not, this is the healing process. Collagen degradation exceeds synthesis. Where does that happen? Degradation. Where does collagen start to break down?
When you're trying to remodel.
Good. So that's the scar, like the third phase, scar
Yeah,
formation.
which is
when you're
I think like
Yeah.
after 1 week.
Yeah, weeks to month, 1 month, right? Around that time. Good. Wound strength is 80% of normal. We said that does happen, but months, like maybe in 3 months' time, according to his example, the doctor's example. Type 4 collagen predominates, not necessarily. That comes, which collagen is it that comes in healing? That's more important.
Type 1.
Good job. Yes, type 1. Okay. Type 4 is collagen. Type 4 is basement membrane. No more wound strength will be gained. No, it will increase in wound strength as you go like week by week after this. So that's why it's wrong. Now, all the answers for these questions are in the notes, but I'll, yes, it's for you guys to kind of like practice on. But yeah, they're in the notes. And I think that's it. Thank you so much for like listening. There's a QR code for your, like if you can fill this in for your feedback. And I really, really hope it helped. My number and email are like on this slide. If you ever need to contact me for anything, if you have questions, just let me know.